Hello and welcome back to the GCRC and Friends web event series. As you well know, we're in the middle of a COVID-19 pandemic, and as we scramble to figure out what this virus does in the epidemiology side and what it means essentially for society, we thought it would be a perfect time to turn to science to make sense of it. And what else better at a resource here in Montreal, in McGill University, than Dr. Selena Sagan. So, Selena, thank you for joining us at this web event series.
Thank you.
Selena is an outstanding researcher here at McGill. She's associate professor in the Department of Microbiology and Immunology, and also professor in the Department of Biochemistry, my own department as well. And she's a renowned researcher in her field. She's Canada Research Chair in RNA biology and viral infections. So as we'll figure out a bit later in our discussion, COVID-19 is the result of an infection by a virus that's an RNA virus. So this virus in particular falls in straight in the expertise of Selena. So Selena, as all the professors in McGill, perform research at the very cutting edge, but also mentor and teach students. So this is the kind of job that we do. Few people realize, right? It's not only research, it's not only teaching, but it's really a continuum of roles. And as we can see here, Selena leads a beautiful group, or a very nice lab right there. And as I understand, most of them are home at this moment, aren't they, Selena?
Yep, that's right. So just like most of the population, we're all undergoing social distancing, and most of us are in isolation. And myself and Carolina, who's a postdoc in the lab, we're working on a COVID-19 related project to try to see if we can effectively inactivate virus on personal protective equipment like masks using UV light, so that the hospitals, which are experiencing a lot of shortages in personal protective equipment, can safely reuse them.
So research on very complicated, tiny details of genetics of a virus, but at the same time, quite practical problems as well.
Yeah, that's exactly it. So we do a lot of molecular virology, but of course we want our work to have practical impact as well on the short term.
Wonderful. So Selena, take it away.
Excellent. Thank you, Thomas. So I thought we'd sort of start at the beginning. So what is a virus? So a virus is an infectious obligate intracellular parasite. And I know that's a large mouthful, so I want to break it down for the audience. So infectious means that it's transmissible, so it can be transmitted from one person to another. Obligate intracellular means that it can only function inside a cell. And parasite is really just what the word suggests. It actually takes advantage of or parasitizes our host cell machinery in order to replicate itself and make more copies. So a virus is an infectious obligate intracellular parasite, or something that's transmissible, that can only function when it's inside a cell and takes advantage of our machinery. And a virus particle itself is actually quite simple. So it's made up of genetic material, so either DNA or RNA, depending on the virus, a protein coat or capsid, and that's, you can see in the cartoon, is in the center here, you can see the thread of RNA, and it's covered in these dots, which are the viral capsid protein, and that's the viral coat. And then in some cases, and in the case of coronaviruses, it also has an envelope that's derived from the host cell membrane. So that's the outside part here of the viral particle in the cartoon. So our cells are used to following recipes. So our genetic material is made up of DNA. You can kind of think of DNA as the cookbook. And the DNA gets transcribed into RNA. And RNA you can think of as the recipe. Now the recipe gets taken up by a machinery called the ribosome. And you can think of the ribosome as the chef. And so the chef takes the recipe and he translates it into dishes, which are proteins. And proteins are what make us. And when a coronavirus infects a cell, it brings with it its own RNA or its own recipe. And the chef or the ribosome of the cell doesn't know the difference between recipes for human proteins and recipes for viral proteins. And so the chef converts the coronavirus recipe into viral proteins and into viral particles.
So it's amazing how these tiny little creatures are not really organisms. They're actually materials, if you will, are actually so efficient that take over the organism and the human body, isn't it?
Yeah, it's quite fascinating.
Another important thing there to mention for both of our labs is that there's a lot of biology and complexity behind the RNA. It's not only a recipe, right? So there's a lot more complexity there, especially in fine-tuning gene expression, but also things like timing of viral infection, but also interactions with the host, hidden in regulation through this RNA part of the material that's presented here. So beyond a recipe, RNA has multiple other purposes.
Yeah, exactly. Yeah. So coronaviruses are a family of large RNA viruses, and they're named for the Latin word crown or halo. And this is in reference to the way that they look like when you look at them under electron microscopes. You can see this viral particle here. It has sort of this crown or halo around it that's made up of the viral spike proteins, which stick out of the viral envelope.
When you
And
look
so
at
that's how.
the way, they're almost pretty.
Yeah, exactly. They're almost pretty, but really deadly, right?
Yeah.
And so that's how they got their name of coronaviruses. And coronaviruses cause respiratory illnesses in humans, and they are also a number of important veterinary diseases that are caused by coronaviruses. And in fact, they're responsible for about 30% of all common colds. So most of us will have had at least one or multiple coronavirus infections already in our lifetimes. And so currently there are no vaccines or treatments available for coronavirus infection, so only supportive care is the only thing that we can do for people that are infected with coronavirus. So SARS-CoV-2 is actually the name of the causative agent or the virus that causes the disease COVID-19, for which there's currently a pandemic. And SARS-CoV-2 stands for Severe Acute Respiratory Syndrome Coronavirus 2. So how does SARS-CoV-2 access our cells? So the coronavirus has a spike protein on the surface, which is this blue protein. And the blue protein or spike protein you can think of as kind of like a key. And it interacts with the lock on our cells or the door to our cells, which is the receptor called ACE2. And the spike protein therefore uses the ACE2 receptor that's on the surface of human airway cells in order to unlock the cell and gain entry into our cells. And so once these two proteins interact, that unlocks the door to the cell, and the virus can enter the cell by a mechanism that's known as endocytosis. And then once it's been entered into the cell, you get a fusion event between the viral envelope and the human cell membranes, which releases the viral RNA into the cytoplasm of the cell. And then the viral RNA can be taken up by the ribosome or the chef and made into copies of viruses.
Yeah, and that's how it takes over the whole kitchen, isn't it?
That's exactly right. Yeah.
Then there's a quite interesting biology behind the ACE2 receptor. This protein actually serves a physiological purpose, something inside our cells. It's not only hanging out there just so we can get infected. ACE2 is an important component in signaling machinery in controlling blood pressure in the cells usually that are either in the lung, but also in the cardiovascular system, and it's distributed in multiple tissues. So the virus can actually infect multiple different tissues, and this receptor actually is something that is normally utilized in our organisms for quite important purposes.
Yeah, that's correct. So receptors aren't just there to catch viruses, they have normal physiological roles in our body. So SARS-CoV-2 is a novel coronavirus. It's actually the third novel pathogenic coronavirus to have emerged in the last 2 decades. And so SARS-CoV-1 or SARS coronavirus was actually emerged in late 2002, early 2003. MERS or Middle East Respiratory Syndrome coronavirus emerged in 2012. And then SARS-CoV-2, the causative agent of COVID-19, emerged in late 2019. And these viruses are zoonotic pathogens, which means that they originated in animals. And it's likely that they originated in bats, possibly through an intermediate host. So for SARS-CoV, the intermediate host is the palm civet cat. And for MERS, the intermediate host is the camel. But the intermediate host for SARS-CoV-2 is not yet clear. So where did SARS-CoV-2 come from? So if we compare the sequence of SARS-CoV-2 with other coronaviruses that we've already knew about, you can see that they actually have 96% identity or similarity to bat coronaviruses, which suggests really that bats are probably a reservoir host, or could have served as the intermediate. And then you can see that there's a reduced relatedness to other coronaviruses that we know of. And so there was a lot of talk early on in the pandemic that perhaps the pangolin was the intermediate host for SARS-CoV-2. However, as you can see, there's only about 91% sequence identity, which sort of suggests that the pangolin is unlikely to have been the intermediate host, at least based on the genome similarity.
Yeah, so in summary, the genetic data suggests that the closest form of the virus is detectable in bats. And that's why a lot of people think that the bats are actually the reservoir. But in that wet market, that in Wuhan, for example, it wasn't obvious exactly where the bats would come from. And I believe it was a period where there's so many bats around. So, and traditionally, there are intermediate species. So it's a remaining question. We don't know at this moment the answer to that as to whether there was an intermediate species or whether it jumped directly from the bat to human in this case. But Selena, I think the lingering question in our mind here is how can a virus actually succeed in jumping between species? How can it actually jump between species this way and change and adapt to new species?
Right, exactly. So how do these viruses make the species jump? And so what can happen is that these viruses, they can make subtle changes, or mutations can accumulate over time in their genomes. And these subtle changes can lead to changes in, for example, that spike protein, which is the key to our cells. And so what can happen is the bat coronavirus could make some subtle changes which allow it to unlock not only the door of the bat cells, but also could unlock an intermediate host or a human host. And then within that intermediate or human host, the virus can undergo some further adaptation, which makes the virus able to infect a larger population of a species like us. And so subtle changes in that spike protein, for example, can alter the tissues that can be infected, the host range that can be infected by the pathogen, and as well as how the disease might manifest itself. So how do these viruses specifically evolve and change over time? And so the viruses carry with them an enzyme called the viral polymerase. And the viral polymerase is responsible for copying the viral RNA genome. And so RNA viruses, such as coronaviruses, they actually make a copy of an antigenomic copy of their RNA genome, sort of like a mirror image. And when they make this mirror image, and then they use this mirror image to actually make more viral RNA genomes. And when they do this procedure of copying, the viral polymerase itself is error prone, so it can make mistakes, and these mistakes can cause changes in the virus. So sometimes these mistakes don't cause any changes, sometimes they make the virus weaker, and sometimes they might make the virus stronger. And then there's a second mechanism by which coronaviruses can accumulate mutations. And so in this mechanism, what happens is you have two viral genomes, viruses that have infected the same cell. So you have the dark blue viral genome number 1, and you have a light blue viral genome number 2. And when the viral polymerase is copying the viral RNA, sometimes it can fall off the viral RNA. And so instead of jumping back on that first piece of viral RNA and continuing to copy, it can actually jump onto a different viral genome in the cell. And this results in a chimeric RNA genome that's sort of half a copy of the first viral RNA and half a copy of the second viral RNA. And this is a mechanism known as replicative recombination. And so these two mechanisms, error-prone RNA replication and replicative recombination, can result in these viruses evolving or changing over time.
So what's fascinating here in case of these viruses is that they have a really different way of regenerating or copying their own genome. Mammalian cells use DNA polymerase. We encode our genome in DNA, and we use a whole different flavor of polymerase to copy our genome. Whereas these viruses use an RNA-dependent RNA polymerase to replicate. And that, as we'll see a little bit later, actually is an opportunity for therapy, because this is a fundamental difference between the host, us, and the pathogen, the virus. And therefore, it stands to reason that it might be a good therapeutic target. And we'll see it actually is a strategy that many of the drugs are utilizing. The other really fascinating part here is that the virus actually benefits its randomness, but because there's so many copies of it, it benefits once in a while from mutations in its genome. So we see essentially as a reproduction of a species as the better, the more reliable the copy is, the better for the species, right, in terms of humans. But in case of viruses, it can be advantageous for mutations to arise randomly in order to generate variety. And the two mechanisms you present here, because of this viral, this RNA-dependent RNA polymerase biology, actually offer, if you will, more opportunities for adaptation.
Yeah, exactly. All right, so how does SARS-CoV-2, this causative agent of COVID-19, differ from other respiratory viruses? So you can see right off the bat, if you look at the cartoons of the different viruses that we've drawn here, that influenza virus, which causes the flu, actually looks very different from SARS-CoV-2. So you can see that influenza virus actually has 8 pieces of RNA in its genome, and SARS-CoV-2 only has 1 piece of RNA inside its viral particle. And so just even the type of genetic material that they have, or the number of strands of RNA that they carry, is very different. You can see the structure of the cartoon of the virion is very different as well. And so one of the important concepts that I want to point out on this slide is the basic reproductive number. So the basic reproductive number, which is also known as R0, is a really important concept in virology. So the basic reproductive number actually is a representation of the average number of cases one case generates in the absence of any countermeasures. So in the case of flu, you see that for every person infected, they infect 1.3 other people. So they usually infect 1.3 other people. Whereas with COVID-19, you'll see this number is much higher. So for every person that's infected with COVID-19, in the absence of any countermeasures, they typically infect 2 or 2.5 other people on average. And so this is actually the reason that we're undergoing these social distancing and self-isolation procedures right now, because what we want to do is introduce a countermeasure that could reduce that number and drive that number down, so that every case of COVID-19 leads to less than 1 other case of COVID-19. And if we can do this, that means if this number is under 1, that means that the infection will die off in the long term. And so that's really the importance of social distancing and self-isolation. I also want to draw a little bit more of a comparison between flu and COVID-19, because these two viruses have been sort of talked a lot about in parallel as being similar, and I think they're quite different, in fact. So you can see that with flu, the incubation time is about 1 to 4 days, whereas with COVID-19, it takes about 4 to 14 days before symptoms arise. And what this means is that there's a longer period of time where patients are infectious, so they can spread the infection, but actually don't have symptoms. So they can spread the infection for a long time before they realize they're infected. And in fact, some patients are actually have quite mild symptoms. So they can spread the infection for a long period of time, but don't know that they're actually infected. And then on top of that, you can see the hospitalization rate for flu is quite low. It's about 2%, whereas about 19% of COVID-19 patients require hospitalization. And this is because it's quite difficult to breathe. And then, of course, that leads to a case fatality rate. So with flu, 0.05 to 0.1% is the case fatality rate, whereas with COVID-19, the case fatality rate is probably somewhere around 3.4%, although these numbers are from March of 2020.
Yeah, it's a bit outdated. They will change slightly, right?
Yeah, exactly. So I think sorry, go ahead.
So I just looking at this, though, we see that COVID-19 lies in a very peculiar place in terms of properties, right? On the one hand, it's not as lethal as SARS and MERS. And those viruses, SARS and MERS, were not, if you will, not successful, because if you kill your host, you have fewer chances to spread. On the other hand, you see that the incubation time here is much longer than the other one. And we also know now that there's quite a bit of the population that is asymptomatic, even though they were infected. So it really lies in a sweet spot in terms of or unholy spot terms of properties of virus, but it's highly successful in expanding and replicating itself without killing its host anyway. And what's missing though here is that the case fatality rate is actually quite unequal depending on the age group, and then we've heard quite a bit about that as well.
Yeah, yeah, definitely. So the big question is, right, how can we stop it? And so certainly we want to take a number of protective measures. So we want to maintain a healthy lifestyle. So you want to make sure you're eating well, you're sleeping well, and you're getting enough sunlight to keep your vitamin D levels good. And then in terms of hygiene, we want to really practice good hand washing technique. We want to avoid touching our face. We want to cough into our elbow and wear a face mask to avoid transmitting the infection to others, particularly when we're in that asymptomatic phase or pre-symptomatic phase of the infection. And then, of course, we want to reduce transmission. We want to drive that basic reproductive number R naught down. So we want to isolate and quarantine, and we want to limit our contact with other people to avoid transmitting the virus to others. And so this is really important because we know that the virus survives in aerosols for about 3 hours, so droplets. We know that it can survive on cardboard for about 24 hours, and we know that it can be stable on plastic or stainless steel for about 2 to 3 days in the environment.
Yeah, we heard a lot about these properties and these measures as we were entering in the pandemic. But now that we reached, or many of countries are reaching or have reached the peak of the infection, we're getting into that long-lasting, or if we call this the so-called dense phase, where people will actually progressively enter, get out of confinement. And these measures don't take lesser importance. Actually, they take a greater importance as we move out of confinement, because the risk for exposure is actually greater out there than if you were to stay home with in quarantine and in really limited access. So these measures are not less important now; they're actually more important, and it's important to double up on these guidelines.
Exactly. So as we start to re-enter into our workplaces and re-enter into sort of society with other people, it's important that we really maintain vigilance and really maintain these hygiene practices and really try to reduce transmission of the virus. So one of the main strategies in which we can protect ourselves is vaccine-based strategies. And so there's lots of vaccines-based strategies that are being looked at right now. And so I'm just going to explain here one of the most simple vaccine strategies, which is a protein-based vaccine. And so in the case of a protein-based vaccine, what you do is you take a part of the virus and you produce in a lab. So you don't take it from the virus itself, so it's not infectious. You actually synthesize just a piece of the virus in the lab and you purify it, and then you administer it to people to train their immune system. And so what this means is that the immune system learns to recognize the viral spike protein, for example, and then if that person gets infected with the virus, the immune system says, oh, this is the spike protein, this is bad, and it mounts an immune response to limit infection in the host or in people.
So vaccines and immunizations purpose always the same: amount a defense, amount a memory of those pathogens without the risk associated with the infection by the active agent. So you get all the benefits and very, very little of the risk associated with it.
And I think you said that Terry is going to be doing a talk on vaccines.
That's right. So upcoming, the next GCRC and Friends over the upcoming week, stay tuned, is from Dr. Terry Bear. He's going to present more about the different therapeutic approaches that are ongoing in the clinical trials. So keep abreast of our publications. The next one will be telling us more about all the different possibilities of therapies for COVID-19.
Excellent. So vaccines are one way to prevent infections from occurring. And antiviral therapies are ways to treat people who have active infections. And so I wanted to talk about today a new drug called Remdesivir that's been a lot in the press recently. So Remdesivir is a viral polymerase inhibitor. So it's that enzyme that's responsible for copying the viral RNA. And Remdesivir is a nucleotide analog. So what it looks like is our normal nucleotides that are in an RNA chain. And so if we take a look at the cartoon at the bottom and we take a look at the inside of the viral polymerase, you see that the viral polymerase is sort of like a slider on a zipper, and it builds the teeth of the zipper as it moves along. And so you can see that it incorporates nucleotides as it moves along the viral RNA to make a copy. And at some point, what can happen is it can incorporate remdesivir instead of a normal nucleotide. And when this happens, it causes a bulge in the zipper or in the chain of the RNA. And so the polymerase can add a couple more nucleotides, but what happens is it gets stuck. And this is a mechanism known as delayed chain termination. So the viral polymerase can no longer continue to copy the viral RNA, and this can inhibit the virus. And so remdesivir has been shown to be more effective if it's given at early stages of infection. And quite excitingly, it's been shown to have limited drug resistance in animal models. And so what this means is that the virus can accumulate some mutations which allow it to resist this drug, but those mutations that allow it to resist this drug actually make the virus weaker. And so that's actually quite good for the long-term potential of this drug.
Yeah, the clinical trials so far, anyway, some of the preliminary work that has been done in different models is quite promising. So we probably we probably aren't done hearing of from remdesivir. Something though that emerged was that it was probably more efficient at the beginning of the infection.
Yeah. So there's currently clinical trials being done by Gilead Sciences, who are the makers of Remdesivir. And some of the preliminary data was reported this past week, and it looks quite promising. I think we'll know more in the coming weeks.
It's a nice cartoon there, Selena. It makes this complex mechanism quite clear. Thank you.
Excellent. So what does the future hold? So zoonotic infections are really going to continue to emerge. And so this isn't the last emerging viral infection that we'll see, and we can't really predict them, in fact. But what we can do is we can know what's out there, and we can be ready to tackle what's out there. And so what's needed for us to be able to tackle emerging viral infections in the future? So first, really, we need research, right? So we need to be able to understand these viruses. We need to understand the families. We want to manage and survey wildlife to understand what's out there. We want databases so that when a new infection appears, we can compare it to something. And we really want to know how we can inhibit these viruses through vaccines or through drug-based approaches or antiviral approaches.
Yeah, so a key issue also that you mentioned in the past is the keeping an eye on the actual reservoirs for those viruses, especially knowing that so many viruses actually originate from bats, for example, that we want to keep an eye on those populations and the viruses that are endemic. But the bottom line with everything that you presented and what strikes me is that where these viruses are, there's more and more of them, more and more actually they are entering in population, and that we've essentially we have a lot of catch up to do in terms of toolkits for not only with antiviral drugs, but also in terms of social health and having a consistent public health policy across countries, because it knows no boundaries, as we've seen very well, right? It's a worldwide challenge.
Yeah. So in addition to research, we really need to be able to have good first responder action. So we need the clinicians that are dealing with patients to recognize that an outbreak is occurring and alert the appropriate authorities, such as governmental agencies and international agencies like the World Health Organization. We also need public health action. So we need to empower our governments to be able to stockpile personal protective equipment, vaccines, to institute travel advisories and geographic travel restrictions, and really be able to take care of our population better.
Science, what we know, orienting policy, it makes only sense, right?
Yeah.
But more research is necessary, that's for sure.
Yeah, I think that's certainly a big piece of the puzzle, right? Is really being able to understand and work with these viruses in the lab so that we can know how to tackle them as they arise.
That's right. So, Selena, first off, thank you very much for taking part to that first web event series. and it was quite enlightening. So thank you very much. I also know that Selena picked references for those of you who are curious about COVID-19, SARS-CoV-2, but also the other viruses. Very often those publications, especially the most recent one, are published in specialized journals. So with a language that's somewhat difficult to decipher for the general public. But Selena has picked also references where the information is also boiled down and summarized for general public. So there are actually really good sites out there from CDC, from the Public Health Agency of Canada, and from great associations like the American Association for Microbiology. But there are also some references that are quite interesting out there for those of you who are courageous to go through a bit of the more detailed science. And I know that Selena has picked one of them. So for those of you who are new with the GCRC and Friends web series, we will keep this list of further reading and complementary resources at the bottom of our video in YouTube and other interfaces. So again, Selena, thank you very much for doing this. And well, keep it up with the great work, and good luck with your team here at McGill.
Thank you so much.